Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The intellectual landscape of late 20th century genetics was one of meticulous, artisanal craft. Investigators, armed with nascent recombinant DNA technologies, pursued individual genes with the dedication of medieval manuscript illuminators, tracing inheritance through complex pedigrees and celebrating the localization of a single locus as a monumental achievement. Into this world of bespoke science, Eric Lander and David Botstein introduced a paradigm of industrial scale and mathematical certainty. Their collective vision, coalescing around the year 1990 and articulated in a series of seminal papers and strategic proposals, can be understood as a singular intellectual project: the systematic and comprehensive mapping of the human genome. This was a prospectus for a new type of biology, one founded upon principles of engineering, statistical rigor, and exhaustive completeness. This essay provides a profound evaluation of the internal claims, arguments, and specific conceptual frameworks presented within the Lander-Botstein vision for genome mapping. It will proceed by systematically analyzing the foundational pillars of their argument, from the initial proposal of a comprehensive linkage map to the strategic insistence on a hierarchical, map-first approach, assessing the coherence of their logic and the generative power of their foundational claims. Section 1: The formalization of a comprehensive genetic linkage map. The first foundational claim, articulated most clearly in the work preceding 1990 by Botstein and his colleagues, was that a complete genetic linkage map of the human genome was a tractable and achievable goal. This assertion represented a profound conceptual leap. The internal logic of the claim rested upon the power of naturally occurring human genetic variation. The central mechanism proposed was the use of what they termed restriction fragment length polymorphisms, or RFLP. These are inherited differences in DNA sequences between individuals that create or abolish recognition sites for specific restriction enzymes. When genomic DNA is digested with such an enzyme, the resulting fragments will be of different lengths for different individuals, a variation detectable through gel electrophoresis and Southern blotting. The claim was that a sufficiently dense collection of these polymorphic markers, distributed across all chromosomes, could serve as a comprehensive set of genomic landmarks. The argument's strength derived from its elegant application of classical Mendelian genetics to the molecular level. Linkage analysis, the statistical method for tracking the coinheritance of a marker and a disease trait within families, would gain unprecedented power. The closer a marker is to a disease gene on a chromosome, the lower the probability that a recombination event during meiosis will separate them. This frequency of recombination, measured in the statistical unit of centimorgans, provides a measure of genetic distance. The claim, therefore, was a statistical one with enough well-spaced, highly informative markers. Any gene responsible for a simple Mendelian disorder could be mapped to a specific chromosomal region with high confidence. The evidence presented for this was primarily theoretical and statistical, a proof of principle that human variation itself held the key to its own systematic deciphering. It reframed genetic variability from a confounding factor into the essential tool for navigation. The evaluation of this claim reveals its deep insight. It correctly predicted that a dense framework of markers would become the primary tool for disease gene hunting for the next decade, transforming human genetics from a process of fortunate discovery into a systematic Section 2: The mathematical imperative for a physical map. Building upon the framework of a genetic map, the Lander-Botstein vision introduced a second, crucial claim: the absolute necessity of a physical map. A genetic map measures distance in probabilistic terms of recombination, while a physical map measures it in the concrete reality of base pairs of DNA. The argument, most powerfully articulated in the mathematical formalisms of Lander and his collaborator Michael Waterman, was that a physical map, composed of overlapping, cloned segments of DNA, was an indispensable intermediate for any large-scale sequencing effort. The proposed mechanism was clone-based mapping. Large fragments of the genome would be cloned into vectors like yeast artificial chromosomes, creating a library of cDNA pieces representing the entire genome. Each clone would then be characterized by a unique fingerprint, for example, the pattern of fragments produced by a particular restriction enzyme, digest. By identifying shared elements in these fingerprints, one could computationally detect overlaps between clones and assemble them into contiguous blocks, or contigs. The claim was that this process, scaled appropriately, could build a scaffold covering entire chromosomes. The argument was distinguished by its rigorous mathematical foundation. Lander and Waterman presented equations that modelled the entire process, allowing for precise predictions about the progress of a mapping project. For instance, they defined the relationship between the number of clones analyzed, the average clone length, and the size of the genome. Their formulas could predict the number of contigs that would form and the probability of closing gaps between them. One central calculation demonstrated that the probability that any given point in the genome remained uncovered by a clone is equal to the mathematical constant e, the base of the natural logarithm, raised to the power of the negative of the coverage ratio, which is the total length of all clones divided by the genome size. This mathematical representation of the project transformed it from an open-ended exploration into a solvable engineering problem with predictable costs and timelines. The claim's evaluation shows its strategic brilliance. It provided a rational, quantitative basis for planning the Human Genome Project, arguing that efficiency and completeness required this intermediate step. The physical map was presented as an essential navigational chart without which any attempt to sequence the vast, uncharted territory of the genome would devolve into confusion. Section 3. The abstractive power of the sequence tagged site. A persistent challenge in larger scale biology projects is the problem of standardization and data integration. Different laboratories produce different results using different reagents, making comparison and synthesis extraordinarily difficult. The Lander-Botstein Consortium, along with Maynard Olson and Leroy Hood, addressed this with a third and particularly elegant claim: the concept of the sequence tagged site, or SDS, as a universal language for genome mapping. An SDS was defined as a short, unique stretch of DNA sequence, typically a few hundred base pairs long, that is operationally defined by a pair of polymerase chain reaction primers that amplify only that specific segment from the entire genome. The claim was that by building a map based on a dense collection of these SDSs, the scientific community could create a universal reference system. The internal logic was one of abstraction. Previous physical maps were tied to physical artifacts, specific clones in a specific freezer in a specific laboratory. These clones were prone to contamination, rearrangement, and were difficult to distribute globally. An SDS, in contrast, is pure information. The sequences of the two PCR primers are all that is needed to identify that point on the genome. Any laboratory in the world with a DNA synthesizer and a PCR machine could recreate the assay and verify a map location for themselves. This claim effectively proposed to dematerialize the genomic map, translating it from a fragile collection of biological samples into a robust, easily transmissible digital database. This was a claim about the fundamental nature of scientific currency. It argued that the most reliable and scalable representation of a genomic map was informational, a sequence of characters in a database, instead of a physical object. The evaluation of this claim highlights its profound and lasting impact. The SDS concept was adopted as the central organizing principle for the Public Human Genome Project. It solved the problem of map integration between different centers and different types of maps, such as genetic linkage maps and physical contig maps. It created a common coordinate system that was stable, reproducible, and democratic, forming the essential informational backbone upon which the final reference sequence was. Section 4. The strategic superiority of hierarchical scaffolding. The culmination of these preceding claims was a fourth, overarching strategic argument: the advocacy for a hierarchical, map-first, sequence-eligible approach to deciphering the entire human genome. This claim was perhaps the most contentious, as it positioned this strategy in direct intellectual opposition to alternative proposals, such as whole genome shotgun sequencing, which proposed to sequence random fragments first and assemble them computationally later. The internal claim of the Lander-Botstein camp was that a hierarchical approach was essential for ensuring the accuracy, completeness, and long-range continuity of the final human genome sequence. The logic was grounded in the known complexity of the human genome, particularly its vast tracts of repetitive DNA. They argued that attempting to assemble billions of base pairs from short, random sequence reads without a guiding map would be computationally overwhelming and fraught with error. Repetitive sequences would act as confounding elements, causing misassemblies where distinct genomic regions were incorrectly collapsed into one, or single regions were incorrectly expanded into many. A high-resolution physical map of SDSs and overlapping clones, they claimed, would provide the necessary scaffold. The genome would be broken down into large, manageable fragments of known order and location, for example, a series of adjacent yeast artificial chromosome clones. Each of these large fragments would then be sequenced individually, and the resulting sequences would be pieced together according to their pre-established order on the map. This method, they asserted, would be analogous to assembling a 1,000-piece puzzle by first looking at the picture on the box. It was a claim about quality control and risk management. It promised a finished product of high fidelity, with all a sequence anchored unambiguously to a specific chromosomal location. Critically evaluating this claim unveils a fascinating chapter in scientific history. The Public Human Genome Project adopted this hierarchical strategy as its central dogma. Later, the private Cellular Genomics, led by Craig Venter, championed the whole genome shotgun method and produced a draft sequence with impressive speed. Yet, the ultimate assembly of the Sarah sequence relied heavily on the publicly available mapping data for scaffolding and validation. The Lander-Botstein claim about the difficulties posed by repetitive DNA was entirely correct. Assembling the shotgun data into a coherent whole proved to be a monumental challenge, and the public map provided the essential long-range framework to resolve many ambiguities. The claim for the strategic superiority of the hierarchical approach was thus vindicated in a complex way by demonstrating that a map was indispensable for achieving the quality and completeness they had originally defined as the project's or Section 5. The generative capacity of mathematical formalism in biological enterprise underpinning all these specific claims about maps and sequences is a deeper, more fundamental metaclaim implicit in the entire Lander-Botstein corpus, that larger scale, biological endeavours are best conceived, planned, and executed as exercises in applied mathematics and engineering. This was a radical departure from the prevailing culture of discovery-driven, hypothesis-testing biology. The work of Lander, a mathematician by training, and Botstein, a geneticist with a deep quantitative intuition, is saturated with a mathematical worldview. Their papers are filled with probabilistic models, algorithms, and statistical projections. They were a prospectus arguing that biology's next frontier required a new kind of thinking. The internal logic of this metaclaim is that the complexity of the genome is a problem of information management and signal processing. By formalizing the process of mapping, as with the Lander-Waterman equations for contig assembly, they make the problem subject to prediction and optimization. They define success not in terms of a single brilliant discovery but in terms of achieving specific, quantifiable milestones. A map with a certain marker density, a physical map with a certain percentage of coverage, a sequence with a defined error rate. This approach claimed that one could understand the logic of the Genome Project through the logic of mathematics even before the biological work was completed. Evaluating this claim reveals it as perhaps their most revolutionary and enduring contribution. They effectively provided the intellectual justification for the creation of bioinformatics and computational biology, the central disciplines within the life sciences. They argued for, and created, a culture in which theoretical modelling and strategic planning were co-equal partners with laboratory experimentation. This claim about the power of mathematical formalism fundamentally altered the sociology and epistemology of biological research. It provided the blueprint for how to undertake massive, data-intensive projects, establishing a precedent for future mixed revolutions in transcriptomics, proteomics, and beyond. Their work asserted that a biologist's most powerful tool could be an algorithm, and a project's success could be charted on a graph of statistical probability. In conclusion, the conceptual framework for genome mapping championed by Eric Lander and David Botstein around 1990 was a masterclass in scientific strategy and persuasion. It was a coherent and interlocking set of claims that built upon one another with rigorous logic. The argument began with the feasibility of a comprehensive genetic map using natural human variation, progressed to the mathematical necessity of an intermediate physical map for scaffolding, offered the sequence-tagged site as a brilliant informational solution for standardization, and culminated in the strategic imperative of a hierarchical, map-first approach to ensure quality and completeness. Underlying this entire structure was a profound belief in the power of mathematical formalism to render a complex biological problem tractable. A critical evaluation of these claims reveals their extraordinary foresight. The framework they proposed became the successful blueprint for the public Human Genome Project, one of the landmark scientific achievements of the era. Their arguments for systematic, larger-scale, and quantitatively driven science permanently transformed the culture of biomedical research, moving it from a cottage industry of individual artisans to a powerful, information-driven enterprise. The legacy of their genome mapping concept is therefore dual. It is both the specific intellectual architecture that enabled the sequencing of the human genome and the more general, enduring demonstration that the vast complexities of life can yield their secrets to the structured, predictive power of mathematical and engineering principles.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.